Starting off with fatty acids, we have to actually understand what a fatty acid is. So fatty acid is broken down by this structure over here. This is a myristic acid. I believe I pronounced that correctly. But over here with all of our fatty acids, we're going to have two characteristics. One of which is going to be this carboxyl group. So I'll put carboxyl group. And as you know, carboxyl groups are going to be polar, which this is going to be hydrophilic. So water loving, so hydrophilic, polar, pretty much the same exact thing. Now over here we have our hydrocarbons, this long hydrocarbon chain right over here, and this hydrocarbon chain is going to be hydrophobic, or non-polar, however you want to go about that. But with all of our fatty acids, we're going to have this carboxyl group and this hydrophobic hydrocarbon chain, and this is going to result in an amphipathic molecule. So amphipathic. So amphipathic just means that we have the polar and non-polar characteristics. Now with our fatty acids, we are also going to have an even number. So I'll put even number of carbons. So you're not going to see a fatty acid that has 15 carbons. We're going to see 14, 16, 18. Now our fatty acids can actually range from 4 to 36 carbons, but if you have less than 14 carbons or you have more than 20 carbons, these are going to be less common. Especially with our plants and animals, we're going to have mostly C16 and C18. This one is going to be C14. We have 14 carbons. But in general, our fatty acids, the most common ones that we have and we're going to study, are going to be from C16 and C18. Those carbon 16s and carbon 18s for, anywhere in our diet that we might include. And with our fatty acids, we have a couple of other characteristics. We can either have a saturated or an unsaturated fatty acid. So saturated basically means that we're saturated with hydrogens. So over here, this is going to be a saturated fatty acid. Saturated. Why is that? Well, because this fatty acid is fully reduced with no double bonds. This is very important. There's no double bonds in our saturated fatty acid. And as you can see, we all have, we have a methylene group going all the way down here into our methyl group down here. This is a stearic acid. So stearic acid is going to be denoted as 18-0 as our sort of notations when we're going through our fatty acids. So this is going to be stearic acid. As you can see, it is saturated. Now, a characteristic of being a saturated fatty acid is that our saturated fatty acids are going to be pretty rigid. So when we see these saturated fatty acids, there's not going to be a lot of movement, a lot of viscosity, I guess you could say. There's not a lot of movement with our saturated fatty acids. Now, what about our unsaturated fatty acid right over here? So we have unsaturated. So how do we know this is an unsaturated fatty acid? Well, right here at C9, we have a double bond. Right over here. Now, because we have the double bond, this is also known as a kink, and because we have this, we have a lower melting temperature, and with our unsaturated fatty acids, we're allowed to be more fluid when this is included, especially when we're discussing our glycerol phospholipids, which are part of our phospholipids. If we have that unsaturated fatty acid, then we may have a more fluid biological membrane, which we'll dive into soon in this video, but basically, one thing you have to memorize with our unsaturated fatty acids is that we have a double bond. We can have 1, 2, or 3, but we are going to have a double bond. Now, there are some characteristics that we have to know. I guess you could say some rules when we're talking about our double bond. First off, with our notation for oleic acid over here, we're going to have 18-1, 1 signifying that we have a double bond, at least one double bond, but then we have the sign delta 9. So delta is going to represent our position, and as you can see, our position for that double bond is at carbon 9. Now, if we had, for example, alpha linoleic acid or alpha linolenic acid and just linoleic acid, then we would have 9-12 and 9-12-15. 9-12-15 is going to be alpha linolenic acid, which is going to be 18-3. But just basically, you have to know that this delta represents that position in which our double bond is seen. Now, with our double bonds, there's three rules. Why did I put a B right there? A double bond has to be in the cis configuration. So as you can see over here, we're in the cis configuration, and just as a review, our cis configuration is going to be when both of our majority groups are on the same side. So as you can see, we have the higher weighted, I guess you could say, group. I'll just say this is the majority group on the top side over here, this is going to be cis configuration. So our double bonds are never in a trans configuration when we're talking about our fatty acids, and our double bonds are never conjugated. So what I mean by this is, like over here, where we have our C9 and C10, you're not going to see another double bond right over here. That's impossible. We're not going to have that with our fatty acids, but instead we're going to have these methylene groups. So we're going to have a CH2 instead. Like for example, if we're diving into, I guess oleic acid isn't the best way of representing this, but if we had linoleic acid, then we would have our double bond 9, 10, 11, 12. So our double bond for linoleic acid is right over here. So I think 9, 10, 11, or be down right over here. So for our linoleic acid, our double bond will be right here. And as you can see, we're just, trying to represent this, but there would be a methylene group between those two double bonds. So it wouldn't be conjugated to where there's a double bond immediately between, there's a double bond right here. That's not the case. Basically, we are going to have a methylene group in between two double bonds, as we see in linoleic acid and also alpha linolenic acid. And our third qualification is our first double bond must be in the position C9 and C10. So our double bond, our first one has to start at that carbon 9 to carbon 10 connection. So these are the three rules that we have to follow with our fatty acids.